Hello everyone, my name is Snehlata Rao and I am a PhD student and a PMRF Fellow at Department of Biological Sciences and Bioengineering, IIT Kanpur. So today we will continue with our second lecture of the chapter Cell, the Unit of Life. But before moving forward, let's have a quick recap of what we studied in our last class. So in our last class, we studied that how does a cell look like. If you remember, the cell basically looks like a compartment or a chamber which has a semi-fluid liquid that is called cytoplasm, a boundary that is called plasma membrane, a genetic material that stores information that was mostly DNA, and some subcompartments that are designated to perform a specific function. And these subcompartments were called organelles. And if you remember, these organelles were either single membrane bound, double membrane bound, or do not have membrane at all. So this is what we studied in the last lecture. We also saw different organelles that were without membranes, double membrane, or single membrane bound. Apart from this, we also studied that cells are of two types. Cells are of two types based on type of nucleus it has, right? So it is divided into two types based on nucleus it has. For example, the first type was prokaryotes or prokaryotic cells, and the second was eukaryotic cell. And we just saw or had an introduction about the prokaryotic and eukaryotic cell differences, that the one has without membrane bound nucleus while the second has well-defined membrane bound nucleus. So in today's lecture, we will study in more detail about one of this type of cell, that is prokaryotic cell. So what is prokaryotic cell? As we have already discussed, it consists of two terms, pro, which means primitive, and the second word, karyotic, that is derived from a word karyon, and it means nucleus. So a prokaryotic cell is one which has primitive nucleus or do not have membrane bound nucleus. Right? They do not have membrane bound nucleus. Apart from this, since these are small cells, we also saw the shape and sizes of different cells, and we saw that the prokaryotic cells are generally smaller than eukaryotic cells. That was in the range of 1 to 5 micrometer. Second, they exist mostly in unicellular form and multiply faster. They multiply faster as compared to eukaryotes. And thirdly, if we talk about its organization, so the organization of these cells are simple because they are mostly unicellular. So they have a simple organization and are fundamentally similar. So it means that these cells have the structure that is almost similar. However, they have various different based on the shape, these are further categorized, which we will discuss later. But if you talk about the examples of prokaryotic cells, so it mainly consists of bacteria. Now, these bacteria can be of two types. The first is eubacteria. So, as the name suggests, it has two terms, eu and bacteria, and the term eu means true. So these are basically true bacteria, such as cyanobacteria. So this consists a class of bacteria that is cyanobacteria. These are also called photosynthetic bacteria. So the cyanobacteria are also known as photosynthetic bacteria, such as blue-green algae. Apart from this, we have another class of bacteria that is known as archaebacteria. So the archaebacteria, these are the primitive type of bacteria or the oldest type of bacteria that is present. So these are primitive bacteria, and these bacteria survives or are generally found in harsh condition. So they are found in harsh conditions, such as in high temperature condition, high salt condition. And why or how they can survive in these conditions is because of presence of a special kind of cell wall. So their cell wall is responsible for their survival in the harsh conditions. If we talk about the example of archaebacteria, so the archaebacteria mainly consist of methanogens. So methanogen are one of the example of archaebacteria. Now, what are methanogens? Methanogens are methane forming bacteria. So we must have studied this term methane in our organic chemistry class, right? So this is an organic compound. So these methanogens make this methane. So these are methane forming bacteria. Now, we have seen that now we have understood that what are the characteristic features of a prokaryotic cells, that they are generally smaller, multiply faster, and have simple and fundamentally similar organization. But if you talk about the types of bacteria, then this bacteria varies in their shapes and functions. And based on the shape, these bacteria are divided into 4 types. So bacteria are 4 types based on their shape. The first type is if a bacteria is spherical in shape. So if it is spherical in shape, it is known as coccus. Second type is if it is rod shaped, something like this. So if it is rod shaped, it is known as bacillus. And this type of bacteria is the most common type. So the most common type of bacteria is bacillus. Thirdly, if it is comma shaped, if a bacteria is comma shaped, something like this, it is known as vibrio. And fourth is if it is spiral shaped, something like this. If it is spiral shaped, it is known as spiralium. So based on the shape of the bacteria into 4 types: coccus, bacillus, vibrio, and spiral. Now, it might also be possible that these cells occurs in a group. So for example, suppose if you see a sample in a microscope and you see something like this, that is 2 cocci are joined together end to end, then these type of bacteria will be called diplococcus. So these bacteria will be called diplococcus. If there is more than 2 cells that are joined together, something like this, or a chain of a cocci, then you will call this bacteria as streptococcus. It will be called as streptococcus. And if it has arrangement something like this, it is arranged in the form of a grapes, so you will call this bacteria as staphylococcus. Okay. So apart from the shape 4 types, the bacteria can also be divided into different types based on the number of cells they have when you see it under the microscope. For example, diplococcus, streptococcus, and staphylococcus. Now, before moving forward, you also need to understand one more term about the prokaryotic cell, that is pleomorphism. So what does term pleomorphism means? It means the ability of a bacteria to change its shape and function. So if a bacteria is able to change its shape and function, then this bacteria will be known as pleomorphic bacteria. And why or how does this bacteria do this? It does this under the influence of the environmental condition. So the pleomorphic bacteria changes its shape under the influence of environmental condition, right? So if you change the environmental condition of a bacteria, it can undergo change in shape and function, and these kind of bacteria are called pleomorphic bacteria. So in this slide, what we have learned, we have seen the basic characteristics of a prokaryotic cell and how does their shape vary, and based on their shape, what are the types of bacteria. And then we understood what does a term pleomorphism means. Now, we'll study that how does a bacteria looks like. So just like in our last lecture, we saw that how does a cell look like. Now we'll see that how does a typical prokaryotic cell looks like. So the typical prokaryotic cell have a outer boundary. So this is a boundary of a prokaryotic cell that is called cell wall. The boundary is known as cell wall. Then after this, the bacteria have a outermost layer which looks something like this. So the outermost layer of a bacteria, it looks something like this. And this outermost layer is known as glycocalyx. This is known as glycocalyx. Apart from this, the bacteria also has an inner layer, just like we studied in all type of cells. So it has an innermost layer or innermost boundary, you can say. And this innermost boundary is known as plasma membrane. So this boundary is known as plasma membrane or cell membrane. And these three boundaries together are known as cell envelope. These three boundaries are known as cell envelope. So the cell envelope or the boundary of a bacterial cell or a prokaryotic cell is made up of three layers: glycocalyx, cell wall, and plasma membrane. Now, in this plasma membrane, there are certain enfolding. For example, there is one enfolding on which generally the enzymes are placed. And this enfolding is known as mesosome. This enfolding is known as mesosome. Second enfolding that a bacteria can have can be a bigger enfolding on which pigments are attached. So on this enfolding, pigments are attached, and this enfolding is known as chromatophores. This enfolding is known as chromatophore. So what is a chromatophore? It is basically, we can break the chromatophore term in two words. First one is chroma and the second one is phore. So the chroma stands for color and phore stands for pigment. So these are colored pigment containing membranes. So this is the structure or the boundary of the cell. Now inside the cell, we have studied that all the typical cell consists of a semi-fluid matrix, right? So this semi-fluid matrix, what does it call? It is known as cytoplasm. Then again, all cells have another typical component that is called genetic material. And in prokaryotic cell, this is known as nucleoid. So it also has nucleoid. Apart from nucleoid, it has certain bodies that is used for storage. These are granular bodies, and these granular bodies are known as inclusion bodies. So the inclusion bodies are used for storage. Apart from inclusion bodies, the bacterial or prokaryotic cells also have certain organ that is without membrane, and this organelle is known as ribosome. We also studied in brief about ribosome in the last lecture, that this organelle is present in both eukaryotes and prokaryotes, and this is without membrane. So this is the overall structure of prokaryotic cell. But these prokaryotic cells also have surface structure, that a structure that is present on the surface apart from the boundary. So one of this structure is the extension of the cell wall, and this structure looks like something like this. So what does this structure is called? This structure is called flagella. So this is called flagella. The second surface structure that a prokaryotic cell has, it looks like tube-like, and this is known as pili. And the third structure that it has is hair-like structure, and it is known as femore. Okay, so these together makes the surface structure. This together makes the surface structure of a prokaryotic cell. But if you ask me that all of these components or all of these structures are present in all prokaryotic cell, the answer will be no. These structures vary from one bacteria or one prokaryotic cell to another prokaryotic cell. However, there are certain common components, a common structure that is present in all prokaryotic cells. And these structures are, the first one is plasma membrane or cell membrane. So the first structure, that is boundary, is the plasma membrane. Second is cytoplasm, that is present in all the prokaryotic cells. The third is the genetic material, that is nucleoid. And the fourth is ribosomes. So these are the four structures that are present in all the prokaryotic cell, irrespective of its type. Okay, so in this we studied what are the different type of structures that is present in a prokaryotic cell. Now we will see these structures in detail one by one. So the first structure that we will discuss is cell envelope. So we'll discuss about cell envelope in the first structure. So if you remember, I just told the cell envelope consists of three parts, right? So a cell envelope has three parts. It looks something like this. Okay, so it has three parts. The outer layer, if we talk about outer to inner, so the outer layer, it is called glycocalyx. The outer will be glycocalyx. The middle one will be cell wall, and the innermost layer will be plasma membrane. So if someone asks you that write down the name of the cell envelope component from outside to inside, so your answer should be it should be glycocalyx, cell wall, and plasma membrane. Now, what is the role or characteristic feature of this cell envelope? So the cell envelope forms a single protective layer. So although it has three layers, but these three layers together forms a single protective layer. It forms a single protective layer. Secondly, these three components are tightly bound to each other. So these three components are tightly bound to each other. And third, although it consists of three different components, and these three components are made up of different kind of chemicals that you will study in later slide. But so these cell envelope is characterized as a chemically complex component, right? So it is characterized as a chemically complex structure or chemically complex component. So we can write the cell envelope is a chemically complex structure because its three layers are made up of different type of chemicals. Okay. So this is what cell envelope function is and how does it look. Now we will study about all the three components of cell envelope one by one. So the first component is glycocalyx. Okay. So the glycocalyx, it is the outermost layer of the cell envelope. And so it is the outermost layer that I have written, outermost layer. Secondly, it is made up of, what it is made up of? It is made up of mucus plus polysaccharide. So it is made up of mucus plus polysaccharide, and hence it is also called mucopolysaccharide. It is also known as mucopolysaccharide. Okay. What is its role? So the role of glycocalyx is to provide adhesion. Since it has mucus, it has this sticky property, right? So it is used to make because of its adhesion property. So it has this adhesion property. Okay. Now, this glycocalyx exists in two forms. So the glycocalyx exists in two forms. So the first form is known as capsule. So how does a capsule look like? The capsule, basically, you have this plasma membrane, cell wall, and then this outer thick layer. This is the glycocalyx. So if someone asks you that how does a glycocalyx looks like, so what you'll see, it is thick. So the capsule. So the glycocalyx, when it exists in the form of capsule, how does it look like? It looks thick and tough or hard. So the glycocalyx looks tough and hard and thick. Now, what is its role? Since it is hard and thick, the bacteria hide itself. Bacteria hide itself inside it. And from what thing the bacteria hide itself? It hides itself from host immune system, right? Because our WBC, they recognize these bacteria or the pathogens and kill it. So the bacteria hide inside the capsule from the host immune system. Okay. So if it can hide from the host immune system, it will increase its disease causing ability, right? So the capsule also helps in increasing the disease causing ability of bacteria. Disease causing ability of bacteria. And thirdly, because it is made of mucus, the capsule provides the sticky or adhesive characteristic to bacteria. So it provides sticky or adhesive characteristic. So if the bacteria wants to anchor or to attach somewhere, it can do it with the help of the capsule because it has sticky or adhesive characteristics. Okay. So this is about the capsule. It looks thick and hard, and because of which the bacteria can hide itself inside the capsule from the host immune system, and which in turn increases the disease causing ability of a bacteria. Moreover, it also provides sticky or adhesive characteristic to the bacteria. Now, the second type of the glycocalyx is the slimy layer. So how does a slimy layer looks like? Basically, this is your cell wall. And the slimy layer will look something like this, a single kind of layer, which is irregular type. So if we see from the figure, what we can write about slimy layer, we can write that slimy layer are thin, right? Slimy layer are thin and sticky. So slimy layer are thin and sticky. And what is the role of slimy layer? So what does they do? They prevent loss of, the function of slimy layer is to prevent loss of water and nutrient from the cell. Okay, so it acts as a barrier which prevents the loss of water and nutrient from the cells. Okay, so the cell envelope basically has three parts: the glycocalyx, cell wall, and plasma membrane, among which we studied in detail about the glycocalyx, that what it is made up of and in what form it exists. Now, the second structure that we will talk about is the cell wall. So I just told that a cell wall is the middle layer of the cell envelope. So it is the middle layer of the cell envelope. So it exists in between the plasma membrane and the glycocalyx. Now, what is its role? Again, it also act as a protective layer. It acts as a protective layer. Now, the question is, how does it act as a protective layer? So, a cell wall, if you talk about a cell, the cell has a cell wall, right? It has a cell wall, and now it has a plasma membrane. And inside plasma membrane, it has cytoplasm. And if you remember the first lecture, I told that the cytoplasm is a semi-fluid liquid, but it mostly consists of water, right? This plasma membrane that we are talking about, they are generally soft. Okay, they are soft in nature. This plasma membrane are soft in nature. This is the cell wall. So, because the cytoplasm mainly consists of water, it can exert pressure on the wall of the plasma membrane, right? It can exert pressure. So once it will exert pressure, because the plasma membrane is soft in nature, its shape will distort, right? But this cell wall, these are hard. So they protect the distortion of the cell shape. Hence, it provides the structural support. It provides the structural support to the cell. So its major function is it provides the structural support to the cell by maintaining its shape. So it maintains the shape of the cell by maintaining its shape. Okay. Now, the third thing, apart from this, you can see that if this pressure become more, if the pressure become more, the cell can collapse because its plasma membrane will disrupt. So the cell will also protect. It also protect disruption or collapsing of plasma or plasma membrane or cell. So the major function of a cell wall is it acts as a protective layer because it provides the structural support to the cell by maintaining its shape, and it also protects disruption or collapsing of the cell. Now the second question is, what does a cell wall made up of? Sir, what does a cell wall made up of? So it is made up of two things, which together is known as peptidoglycan. Okay. So if you break the term peptidoglycan, it consists of two terms, peptide, right? It consists of peptide and glycan. So what is peptide? It is basically a chain of amino acid, right? So it is chain of amino acids, and glycans are generally sugar. Glycans are sugar. Now, the cell wall of the prokaryotic cell is made up of two types of sugar. What are these two types? The first one is NAM. What is NAM? NAM is N-acetyl muramic acid. Okay. So the one sugar is N-acetyl muramic acid, and the second type of sugar is known as NAG. It's called NAG. You should remember both of these names because these are important. So the second type of sugar is NAG, that is called N-acetyl glucosamine. So what is N-acetyl glucosamine? It is a modification or modified glucose. It has glucose in its name. So it is a modified glucose sugar. Okay. Now, how does the structure of a cell wall look like? So here I have drawn this. So the red dot here, red dot as representing, say, NAM, and this blue dots are representing NAG. Okay. So these NAM and NAG, that is the sugar moieties, are arranged parallel to each other. Are arranged alternatively. And this peptide chain is arranged or is attached to this NAM. The peptide chains are attached to the NAM, something like this. So it are being attached over NAM. And apart from this, these peptide chains are also cross-linked to each other. So there is this cross-linking. Okay, there will be this cross-linking. And because of this cross-linking, the structure of a cell wall looks something like this. So it looks like a cross-linked structure. So how does a cell wall look? It has this cross-linking structure. So now if someone asks that what is responsible for this cross-linking? So the cross-linking is caused by peptide or the protein chain. So the cross-linking of cell wall is caused by chain of amino acids. Okay, chain of amino acids. So this chain of amino acids are responsible for cross-linking, and which leads to the cell wall formation. Now, you all must have studied antibiotic, that is penicillin, right? You must have heard. It is the first antibiotic to be discovered. First antibiotic that was discovered. So the first antibiotic that was discovered is penicillin. Now, what does this penicillin do? This penicillin prevent cross-linking. So whatever cross-linking is happening by this chain of amino acid, it prevents cross-linking in the cell wall. So if the cross-linking won't happen, the cell wall formation will not take place. So basically, what does penicillin do? It prevents cell wall formation. It prevents cell wall formation, right? So we can see we can say that if someone having a bacterial infection, we can give them penicillin because the penicillin will prevent the cell wall formation and hence the bacteria will die. But does it mean that all the prokaryotic cells consist of a cell wall? No, because we have studied this term mycoplasmas. These are also the prokaryotic cells, a type of bacteria, which are the shortest cell. So the mycoplasmas are the shortest cell, and these cells do not have cell wall. So the mycoplasmas do not have cell walls. So they will be they are insensitive to penicillin. So this antibiotic penicillin or any other antibiotic that disrupt or that prevent the cross-linking or prevent the cell wall formation will not work on the mycoplasma. So if someone is having a mycoplasma infection, they must be given any other antibiotic, right? Because the mycoplasmas do not have cell wall and hence they are insensitive to penicillin or any cell wall preventing antibiotic. Okay. So in this lecture, in this slide, what we understood the major function of the cell wall and what are the components which together makes the cell wall. Now, it has been mentioned in the NCERT that the bacteria are divided into two types. Bacteria are divided into two types. On what basis? On the basis of thickness. On the basis of thickness of cell envelope they have. So I just told what is cell envelope. Basically has three components. So the bacteria are divided into two types on the basis of thickness of cell envelope they have. And what are these two types? These are the first one is gram-positive bacteria. The first one is gram-positive bacteria and the second one is gram-negative bacteria. Gram-negative bacteria. So based on the thickness of the cell envelope, the bacteria are divided into two types. Similarly, we saw in the last slide that based on the shape of the cell, the bacteria are divided into four types. So there are different criteria on what basis a bacteria is divided. Here, they are divided on the basis of the thickness of cell envelope. Okay. Now, who came up with this technique or who divided the bacteria into these two types? There was one scientist named as Christian Graham. Okay, the scientist named Christian Graham. He divided the bacteria into two types by using a technique. He developed a technique, and this technique is called gram staining. So using this gram staining technique, he divided the bacteria into two types. Now we will see what happens in the gram staining. Okay. So in the gram staining, first of all, we need to take a sample, any sample, suppose a soil sample over the slide. So in 9th or 10th during your practical, you must have seen a slide that where you keep certain sample and visualize it under the microscope, right? So on the slide, you take a sample and you spread this sample, okay? You spread this sample in a very thin layer. That thin layer is known as smear, okay? After spreading, you add a dye over it. You add a dye and this dye is known as crystal violet. So the name of the dye is crystal violet. You stain it with the dye. So you stain the sample with a dye. You have stained it. Now, after staining, you add another chemical that is known as iodine. So why do you add iodine? So this iodine, it basically fixes the dye. Iodine is used to fix the dye. Once you have added the dye, now you will add. So you have fixed the dye, okay? Dye is being fixed over this bacterial sample. Now you will add a destainer that we basically use alcohol. So alcohol is a destainer that you will use. And this, after washing or staining it with the alcohol, you will add another component that is known as, you will add another dye that is safranin. You will add another dye that is safranin, and that you will add another dye that is safranin, and then you will visualize it under microscope. Visualize it under microscope. So what we have done here, we have first taken a sample. We have made a thin layer of it, the sample that we call smear, and then stained that sample with a crystal violet dye. It is a dye. After staining it with a crystal violet, we have added another chemical that is iodine, and this iodine is used to fix that dye so that the dye get properly fixed on the sample. After that, we used another chemical that is alcohol, and then after adding alcohol and washing it with the alcohol, we added another dye. This was a crystal violet was the first dye, and the safranin is the second dye. We added safranin, and then we visualize the sample, the slide, under the microscope. So when you visualize it under the microscope, you will see two types of cell. So what are these two types of cell? The first type of cell is the one that retains the crystal violet dye. Okay. So the first type of cell will retain this crystal violet dye, and these cells will look violet or purple color. It will look violet or purple color, and these cells are called gram-positive cells. Okay, these cells are called gram-positive cells. Now, the second type of the cell will be one that will retain the safranin. Okay, that will retain the safranin dye because you have added safranin in the end. So these are the ones that do not retain the crystal violet. So you can also write they or do not retain the crystal violet. Do not retain crystal violet, and these will appear pink in color because safranin is pink in color. So they have not retained the crystal violet but retained the safranin, so they appeared pink color. And these bacteria are called gram-negative bacteria. So the first one is gram-positive cells or bacteria, or the second is gram-negative bacteria. Okay. So based on their ability to retain the crystal violet dye, the bacteria are divided into two types: the gram-positive bacteria and gram-negative bacteria. Now we will see that why does one type of bacteria retain the this crystal violet dye and the other type does not. So for that we will first have to draw the structure of the gram-positive bacteria and gram-negative bacteria. So in the case of gram-positive, we talk about gram-positive bacteria. So the gram-positive bacteria have a structure like something like this. So it have a cell wall. Inside it, it has a plasma membrane, and this cell wall is thicker. Okay. So I just told in last slide that what does a cell wall is made up of? It is made up of peptidoglycan, and so it has this thick layer of peptidoglycan or thick cell wall. Okay. So it has a thick cell wall. This is cell wall of gram-positive bacteria. So it has thick cell wall. A thick cell wall, it means it has high peptidoglycan, right? It has high peptidoglycan concentration. It has high peptidoglycan content or concentration. So this is gram-positive bacteria. Also, peptidoglycan is also known as, so the peptidoglycan is also known as murine. So if the question comes, it can ask either the high peptidoglycan or murine. So you should remember that peptidoglycan is also known as murine. Okay. So a gram-positive bacteria has thick cell wall because it has high peptidoglycan content or high murine content. Now, if we talk about the gram-negative bacteria, so how does a gram-negative bacteria looks like? So if we draw the structure of a gram-negative bacteria, this is the cell wall. This is inside it is a plasma membrane, and it also has one more layer above it. Okay, one more layer above it, and this layer is made up of lipid, and it is called lipopolysaccharide layer. So this layer is called lipopolysaccharide layer. Okay, and this is your peptidoglycan layer. This together forms the cell wall. So this is peptidoglycan layer, and these together form the cell wall, okay? And the inside is plasma membrane. So in the gram-negative bacteria, if you see, the peptidoglycan layer is thin, right? So the peptidoglycan content in gram-negative bacteria is low. So peptidoglycan or murine content in gram-negative bacteria is low. It is around 10 to 30%, okay? But in the case of gram-positive bacteria, it's 50 to 80%, okay? Now, the first difference we saw is in terms of peptidoglycan content. The second difference that we saw is in the term of lipopolysaccharide. So if you see, the gram-negative bacteria have lipopolysaccharide. So gram-negative bacteria have lipopolysaccharide or LPS. Okay, but gram-positive bacteria, they lack lipopolysaccharide or they lack LPS. Okay, so these are the two major differences between gram-positive bacteria and gram-negative bacteria. Okay, so now the question is, why does the gram-positive bacteria retain the crystal violet color and why does the gram-negative bacteria don't? Because you must have seen in the last slide, right? In the last slide, you have seen that we have added alcohol. After staining it with the crystal violet, we have stained our sample or washed our sample with the alcohol. Now, what does this alcohol do? The alcohol is organic solvent, right? Alcohol is organic solvent. And we have studied in our organic chemistry class that organic solvent dissolves organic molecules. And the lipopolysaccharide, consists of lipid. So it consists of organic molecule. So when you add the alcohol or wash the sample with the alcohol, the bacteria which have this lipopolysaccharide layer, it will wash or dissolve this lipopolysaccharide. Hence, the color, whatever color that was present on this layer, will wash off, right? Because of this reason, the gram-negative bacteria are not able to retain the crystal violet color because they are being washed by using the alcohol. While the gram-negative bacteria, because it does not have lipopolysaccharide layer, they retain that crystal violet color. So, because alcohol is an organic solvent, it dissolves this LPS. It is dissolved LPS, due to which this gram-negative bacteria do not retain or cannot retain crystal violet. Okay, so they cannot retain crystal violet dye, and because of this, they appear pink in color when seen under the microscope, while the gram-positive bacteria appears violet or purple in color. So this is what we saw about the difference between the gram-negative and gram-positive bacteria. Now, the next topic or the next structure that we will learn is plasma membrane. So we are seeing plasma membrane. We are reading about plasma membrane since the starting of this lecture. But now we will see in more detail that how does a plasma membrane looks like and what is its major function. So before describing the function of plasma membrane, I'll draw the structure of plasma membrane. So we have seen till now that plasma membrane appears like a boundary, something like this, which has this cytoplasm in it. But if you zoom in or if you see, the plasma membrane actually looks something like this. So it looks something like this, okay? So it has two tails. It is made up of a moiety that has two tails and one that is attached to one head. Now, what does this moiety is made up of, or what this is called? So these moiety are called phospholipids. These are called phospholipids. Again, the phospholipids have two terms, phospho and lipids. So phospho means phosphate, right? Phospho means phosphate, and lipids, that is lipid, right? So it has two components, phosphate and lipid. this phosphate, the head that we are looking, this circular thing, these are phosphate groups. These are phosphate. And the tail that we are seeing, these are chain of fatty acid that makes lipid. Okay, so these are chain of fatty acids that make lipid. So plasma membrane is made up of phospholipid. So if someone asks what the plasma membrane is made up of, so it is made up of phospholipids, right? Okay. Now, the second thing that we are seeing in the structure is what? That it is bilayer, right? So lipids are bilayer. So plasma membrane is, or cell membrane, plasma membrane or cell membrane is lipid bilayer, okay? Because it consists of two layers of lipids, this phospholipids. Now, what is the role of this plasma membrane? So this plasma membrane is making the boundary for the cell, right? So it is preventing the direct interaction from the outside environment with the what is present inside the cell. So it acts as a membrane, or it acts as a barrier, right? But this barrier also allows passes of certain molecules or certain essential chemicals that are required to go inside or outside of the cell. So hence it acts as a semi-permeable membrane. Okay, it acts as semi-permeable membrane. Why it is called semi-permeable? Because it selectively allows certain chemicals or molecules to move either inside or outside of the cell. Now, one of the most important thing that we have seen, but we should write here, that this plasma membrane is present in both prokaryotes and eukaryotes. So we also studied in brief that what is eukaryotic cell. So this plasma membrane is important and present in all prokaryotes and eukaryotes. In all prokaryotes and eukaryotes. Okay. So there's no cell that do not contain a plasma membrane because it is important for both prokaryotes and eukaryotes. Okay, so we have also studied in brief that what is plasma membrane, what it is made up of, and what is its function. Now, one more thing that you should know is the plasma membrane consists of this lipid bilayer, right? So it has phospholipids. So how does these lipids are attached to each other, or how do they form such a compact structure? What forces are used here? So this phospholipids interact with each other by mainly by hydrophobic interactions. Okay, so you should remember that this plasma membrane interact with each other by hydrophobic interactions, because these lipids are hydrophobic in nature. Okay, so they repel the water, they do not attract water. So if they have to interact with each other, they interact via hydrophobic interactions. So what we understood about the plasma membrane, that it is made of phospholipid and it is selectively a semi-permeable membrane that allow exchange of molecules inside or outside the cell. And these lipids interact with each other via hydrophobic interactions. Now, the next thing that we will learn today is about mesosomes. So as we draw the structure, I was drawing the structure of the cell membrane, prokaryotic cell. You see that the prokaryotic cell has certain infoldings, right? So one of that infolding is called mesosome. So what is the function of this mesosome? So let me draw here. So this is plasma membrane, and this has this infolding, right? So this is plasma membrane, and there is this infolding. That is called mesosome. Now, what is the role of this mesosome? First of all, it is the extension of plasma membrane, because it is arising from plasma membrane. So it is extension of plasma membrane, and it exists in three forms. So what are the three forms in which the mesosome can exist? It can exist in three forms. The first one is vesicle. The first form is vesicle. Second is in the form of tubule, tube-like structure. And third is in the form of lamellae. So the extension of this plasma membrane, that is called mesosome, exists in three forms: vesicle, tubule, and lamellae. Now we will discuss about what is the role of this mesosome. So the mesosome have multiple roles, because if you see prokaryotic cells, the prokaryotic cells are simple in organization, right? They do not have much organelles. They do not have any other subcompartment that perform specific functions. So most of the functions that a prokaryotic cell perform, it either happens in the cytoplasm, or these membranes, plasma membranes, modify themselves in certain shapes which can perform this function. So a mesosome perform multiple functions, such as, first one is, it increase the surface area. It increase the surface area for respiration. So all cells respire, right? So the mesosome, because of this infolding, increases the surface area for respiration. Secondly, there are certain enzymes that are attached to this mesosomes and hence are useful to catalyze various reactions. So these mesosome contain enzymes. Thirdly, these mesosomes also help in secretion. So if a cell want to secrete or send something out of its body, so the mesosomes help in these secretions. Fourth, it also contains enzyme that is responsible for cell wall formation. So mesosomes also help in cell wall formation. And apart from this, it also helps in DNA replication. So it also consists of enzymes that helps in DNA replications and cell division. So mesosome perform multiple functions, such as they increase the surface area for respiration, contain enzymes, have role in secretion or sending something out of the cell, contain enzyme for cell wall formation, DNA replication, and cell division. Okay. But one thing that is important and that should be noted is that these mesosome does not play any role in photosynthesis. Okay. So if someone asks that does the mesosome contains enzyme for photosynthesis? No, the mesosome do not contain any enzymes or do not have any role in photosynthesis. Okay. Apart from mesosome, we also saw another extension of the plasma membrane. Right. There was one more extension that was bigger. That was bigger in shape. Right. That was something like this. It was bigger in shape, on which there are different pigments attached. And this structure was what that structure was called. That was called chromatophore. If you remember, that was called chromatophores. So what does this chromatophores do? These chromatophores are, first of all, these chromatophores are again the extension of a plasma membrane. They are again the extension of plasma membrane, just like mesosomes. But they are present exclusively in photosynthetic bacteria. Okay. So the mesosomes do not have any role in photosynthesis, but chromatophores are present only in the cells that do photosynthesis. Okay. So these are present in photosynthetic bacteria. Okay. And if you talk about what are the examples of these chromatophores, so there are certain examples such as nostoc and avelum. Okay, so these are the certain bacteria in which chromatophores are present, and hence the chromatophore has role in photosynthesis because it is present in photosynthetic bacteria only. So I have role in photosynthesis. Okay, so we have studied 5 structures. What is the components of cell envelope? So what does it contain? It consists of glycocalyx, cell wall, and plasma membrane. And then in the plasma membrane, we also studied about the extension of plasma membrane, that is mesosomes and chromatophores. Now, we will discuss certain questions and then we will end this lecture. So the first question is, which of the following structure is not found in prokaryotic cells? So we have drawn the structure of a prokaryotic cell. So among this, which of the structure is not present? Nuclear envelope, mesosome, ribosome, plasma membrane. So the correct answer will be nuclear envelope, because prokaryotic cells do not contain membrane bound nucleus, right? They do not contain membrane bound nucleus. So these nuclear envelope are absent in prokaryotic cells. The second question is, what kind of organelle prokaryotic cells have? So does a prokaryotic cell have single membrane bound organelle, double membrane bound organelle, without membrane, or do not have organelle at all? So if you remember the structure of a prokaryotic cell, we saw that the prokaryotic cell consists of ribosomes. Okay, it consists of ribosomes, right? And these ribosomes are what? These are without membrane organelles. So the correct answer will be without membrane organelle. Now the third question is, what are the major interactions responsible for stabilizing plasma membrane? So we just saw that the plasma membrane is made up of lipid bilayers, right? It is made up of lipid bilayer, and these lipids interact with each other. With what force? They interact with each other via hydrophobic interaction. So the correct answer will be hydrophobic interaction. So that's all for today. We'll discuss more about the structures that are left in our next class. That's all. Thank you.